Welcome everyone to the Third Coast Water Seminar Series. My name is Elena Harkness. I'm the Executive Director here at Current, and on behalf of all of our Third Coast Water Seminar partners, Northwestern University, Argonne National Laboratory, the University of Illinois at Chicago, University of Chicago, and University of Illinois, Urbana-Champaign, I'm really pleased to welcome you here to this celebration of water leadership in research across disciplines and across many different places in the country and the world. This is our monthly opportunity to come together and highlight a topic of interest in water and a researcher that's leading the way, helping increase our understanding of that. Today, we are very fortunate to have Dr. Valeria Molinero from the University of Utah joining us. But just in case it's your first time here at the Third Coast, I'll give you a few logistics items up front. We love this to be interactive, so please submit your questions either in the Q&A tab or in the chat. And also, please feel free to drop your name, organization, location in the chat so we can see who's joining us today. We encourage you to meet each other and connect with other participants. This is the networking that we love to encourage here at Current, so please feel free to use the chat liberally. This event is being recorded, and the recording link will be shared on Current's event page and our YouTube channel, and we'll also send it out to all the attendees post-event. So especially as we transition into a world that includes both virtual and more in-person events again, definitely feel free to share these links out. We find that we get a lot of traction on our YouTube channel post-event. It's a great way to share the content that we're able to bring to you here at Third Coast with people who might not have been able to join us here live today. So our agenda will be pretty straightforward. I'm going to turn it over in a moment to my colleague Seth Darling, who's the director of the Center for Molecular Engineering and senior scientist at Argonne National Lab. He is going to introduce our featured speaker today. She will speak for about 40 minutes, and then we'll spend about 10 minutes in Q&A. So make sure you're sending your questions ahead in the chat. I'll moderate those and make sure we get to as many as we possibly can before we close right at the top of the hour. So without any further ado, I want to turn it over. Oh, actually, no, I'm sorry. Out of practice, it's been a month. I want to give you a little bit of information about Current, the convening organization for Third Coast. So Current is Chicago's water innovation hub. We've been around since about 2016 and have a three-part mission: grow an inclusive blue economy, help accelerate innovation and adoption of water technologies, and solve persistent water challenges. We're here because we all know that we need more healthy water to drink, less flood water in our basements, and share a range of other pressing water challenges here in Chicago, where we're headquartered, but also all around the world. We know that water is one of the defining needs that we have in humanity, and also will create some of the biggest challenges in the next years and centuries to come. We also know that there's big opportunity in water, that we can grow the number of blue jobs, putting Chicagoans and people all around the world to work, and building companies that are solving water challenges, both here in our region and around the world. So our problem statement, as we like to frame it, is that we're not rallying fast enough behind those new ideas and technologies that will help protect health and the environment. Water technology innovation is challenging. Often the problems we're tackling are too large or complicated for any one organization or company to tackle by themselves. We need collaboration, we need partnerships, and Current was purpose-built to help create the kinds of partnerships like the Third Coast represents today among research institutions, their industry partners, the community organizations that are close to some of the challenges we're trying to address, all of us trying to help move this farther, move this faster, to help take on water challenges and tackle them in ways that allow us to de-risk them and bring them to life in the real world. So we do that by being an unbiased advocate. We try to serve a search and scout function, staying on top of the latest in water technologies. Third Coast is one way that we do that, leveraging our research network here. We also provide independent assessment and try to help others figure out what are the best technology solutions to deploy, whether it's in industry or with a community partner, or for us to pilot ourselves. For an example, here in Chicago, we're about to launch our second season of Insight real-time water quality monitoring here in the Chicago River. That's a really exciting partnership with the Metropolitan Water Reclamation District of Greater Chicago, City of Chicago Department of Water Management, a number of technology innovators, our research partners here. None of it would be possible without their support, but Current is the hub that brings that together. So stay tuned for h2nowchicago.org to launch later this spring. We also have an emerging pilot portfolio tackling some of the most emerging challenges on the radar, sewage surveillance and using wastewater as an early warning indicator for COVID-19 recently, but also other diseases as a public health management tool. We're working on nutrients and managing nitrogen and phosphorus in our waterways, on the intersection of wastewater and energy, on PFAS elimination. So many different opportunities to think about how to bring technology to bear on these challenges. So without any other preamble, I want to kick it over to our very good friend and partner, Seth Darling, to introduce our speaker, Dr. Valeria Molinero. Seth, coming to us from the airport, and hopefully, let's see. All right, can we hear you?
I hope so, yeah. Hi,
perfectly.
Elena. Thank you.
Right.
So it's my pleasure today. First of all, apologies for the background noise, but it's my pleasure to introduce Valeria Molinero. Valeria got her PhD at the University of Buenos Aires before becoming a postdoctoral researcher at Caltech. She then had research positions at Arizona State and back at Caltech before moving to her current institution at the University of Utah, where she is currently the Jack and Peg Simons Endowed Professor of Theoretical Chemistry and Distinguished Professor. She is also the director of the Henry Eyring Center for Theoretical Chemistry. Valeria's research focuses on understanding and controlling phase transformation and dynamics in materials, with a particular interest in water, which is why we're so excited to hear from her today. And Valeria has been recognized with far too many awards for me to list, but a few highlights are the Beckman Young Investigator Award, the Camille Dreyfus Teacher Scholar Award, the Cozzarelli Prize from the Proceedings of the National Academy of Science, and she was recently elected to the American Academy of Arts and Sciences. And to preserve as much time for Valeria as possible, I'll stop right there and turn it over to her.
Thank you, Seth. Thank you, everyone, for coming here, and I'm looking forward to the discussions afterwards. It's my pleasure to be presenting to you today a work we have been doing in the last few years on understanding ice nucleation and also interaction of biological molecules with ice. And when we see this picture, that this is a picture of Alta here, close to my home in Utah, you see a lot of snow. And of course, everyone knows that the snow comes from clouds. And maybe what is not so obvious is that in the clouds, not only the snow originates on the formation of ice, but also the formation of ice is the leading cause of precipitation in the liquid form. So when we look at a cloud, and we see a cloud that goes up in the atmosphere, what we know is that the higher the temperature, the higher the altitude of the cloud, the colder is going to be the cloud. And there are two temperatures that are important on looking at clouds and seeing what is the state of water there. The state of water in clouds that are above 0 centigrade, the melting point of ice, so it can be a mixture of ice and liquid. Now, if the temperature goes below the melting point, between 0 centigrades and minus 40 centigrades, that is the temperature of homogeneous nucleation, and this means that is the temperature at which any cloud droplet spontaneously will form ice without any help, what you have is that some of the particles, if you go in between these two, you have to really understand what is the structure of each of the droplets in the clouds to predict whether the cloud is liquid or ice. And this is the regime between the temperature of melting and the homogeneous nucleation, where the temperatures are such that some of the cloud droplets are going to have particles that favor the crystallization and they are forming ice, and some of the cloud droplets maybe have no particles, or maybe they have particles that do not help them form ice. So what you have is what is called a mixed phase cloud, and these mixed phase clouds, what they have is that some particles are going to start forming ice, and they're going to extract the vapor out of the liquid particles, and these particles grow. And the growth of these particles in the mixed phase region is what really originates ice particles that are grow enough to finally grow enough to fall from the sky, and in this falling they produce precipitation. And almost all the precipitation that we see as liquid or ice, except in the tropical regions, originate in ice formation. So this idea, this concept of the formation of ice in the clouds as initiation of snow and ice and rain has been known for about a century. But this idea was not exploited to produce precipitation in an artificial way until really the end of the Second World War. So at that point, after the war was over, there was a lot of research done before and during the war on atmospheric sciences. And one of the spin-offs of this was work done in General Electric in the group of Langmuir, that he's a Nobel Prize known for his work on surface science, but he also had a group doing a lot of different interesting things at General Electric. And one of the experiments that they did is they were doing what is called a cloud chamber. And in cloud chamber, you have low temperature vapor. And what they did is they added to this dry ice, the one that we use to cool, that is just solid carbon dioxide. And this dry ice produced the formation of a cloud. And this cloud produced crystals, and this precipitated. So this really captured the imagination of these scientists, Langmuir, Vonnegut, and Schaeffer. And already by the end of 1946, there were the first episode of cloud seeding for real. So Schaeffer went with a small Cessna to one place in New York, close to General Electric, and they put about 6 pounds of CO2 in the atmosphere in some clouds, and they managed to produce precipitation. And what was important about this experiment, it was not so much that it was a very successful experiment, but they attracted the interest of the military that had been working with Langmuir for some time. And the military became involved, and they started supporting these experiments and paying for it. And the main origin of the interest of the military on getting into this area of cloud seeding, so clouds, to seed the clouds to produce precipitation, was on one side the idea of expanding agriculture to regions that may be water scarce, like Utah, for example. And this was not only in the US, but also in other countries. The Soviet Union was doing the same. And the second one was actually pushing all this forward, were military interest. So there was an interest on using control of the weather as a weapon. And I don't mean that you use a weather weapon, like there's a weather that explodes, but mostly that you can control fog, you can control precipitation, and you can control storms and redirect storms. And this could be used in the context of war to really have a tactical advantage. So there was a lot of investment in this area. And actually, as you know, when there is investment in one area, there is a blooming of research in this area. And this was evident in ice nucleation. For example, the first paper on ice nucleation, on this heterogeneous ice nucleation, was the nucleation of ice by silver iodide. And this was done by Vonnegut, also in the previous picture, that was working on developing better nucleants. And what he did is he investigated all the possible structures of crystals that were known in 1947, or 46 actually, and he found that silver iodide has a structure very similar to ice, and he tried it, and actually it's a perfect ice nucleant. By perfect, I mean that it's very good at nucleating ice at very low supercooling, very close to the melting point. Then there were more work, in this case in the Soviet Union, to introduce ice nucleation, to do ice nucleation to control fog and other meteorological events by using organic crystals. And I pinpoint to this paper because it's the first one that they use organics to produce formation of ice. And from there on, there was a big push, and there were plenty of papers. And this is one review, for example, by Fukuta in 1965, in which already 329 organic compounds have been tested for ice nucleation, and more than 20 were found to nucleate above -5. That is as good as silver iodide and pretty good to form ice nucleation in clouds and induce precipitation. And you see that the idea is to use this as a seeding agent for clouds. And all this, of course, it appears also in the popular literature. Maybe several of you have read this paper, this book, this novel by Carl Vonnegut, that is the brother of Bernard Vonnegut that was working on ice nucleation. And in this book, there is a mad or sort of a spineless scientist, and actually it seems to be modeled on Langmuir. And the idea is that this scientist has made a substance there is called ice-9, and this substance is so good at forming ice that can freeze water at any temperatures, and essentially is the doom, is an ecological catastrophe because all the water on Earth ends up freezing. But this idea of making something that is so good that can freeze water at any temperatures, actually, it has some sort of correlation with results that were coming at the time. And by 1967, there was a paper, and then followed by some other papers by the same authors, in which they show that some organic crystals can nucleate ice, they can produce the formation of ice even without any supercooling. So you don't have to go to -2, -3, -4. Already just below the melting point, you can form ice. So there were a lot going on. And actually by 1972, more than 1,000 organisms have been tested and 47 were found to be good for cloud seeding. And if you're in the area of cloud seeding, you may see why people are still using silver iodide and why there is not much more research that has been known in this area. This is not something that is much well known. And the reason has to do with the original enterprise for which this was funded. I was telling you that the funding in this area mainly came from the military, although there was also commercial enterprises. And this is because the military started using cloud seeding for military operations already in the 60s. When it was the Vietnam War, there was an operation that is called Popeye, in this case, that was running from the Nixon administration until 1972. And the idea of that operation was to seed the clouds in the path of the Vietcong to produce an excess of rains, and this would muddle the supply route of the Vietcong and difficult their resupply. So this idea that, of course, as you can see, didn't work very well in terms of the war, and also the Vietcong was able to really go with bikes through this area, was a secret operation, but it came out in 1971, and then in 1972, with the leaking of the Pentagon Papers, it became more evident that the US was doing weapon warfare based on weather. And the US Senate, there was an outcry, of course, of the public, and the US Senate called for a treaty to ban weather modification as weapons that was finally signed 5 years later. But the main implication of this turmoil associated to the use of weather modification as a weapon was that all federal funding in this area essentially dried out in the early 70s. And the only operations that continue some research on cloud seeding were moved to the States. And that means that in some sense, the fundamental science associated to ice nucleation and cloud seeding stopped in the 70s and didn't move forward. This is quite evident when we look at the papers that are more cited per decade that have to do with ice nucleation. I looked at these last year, so some of them are a little bit outdated. But you can see the story in 1950s, the most cited paper that arguably doesn't have too many citations is one on nucleation theory. It actually is a fantastic paper. And this one is on the fundamentals of ice nucleation. Then the same in the 1960s, they're more on ice nucleation and interfaces, but all the work is on the fundamentals. And already in the 1950s and 60s, a lot of the work was shifting towards cloud seeding. All the idea was to find better nucleants that you can use to seed the clouds and control precipitation. However, after the debacle of the Operation Popeye and the ensuing outcry of the public, actually the funding, the research, everything in this area sort of stopped. And then you can see that the most important papers coming out after that are moving into biological ice nucleation. And this started with the finding that the leaves of many plants were able to form ice at temperatures that extremely high, like -2 centigrades. And this was found to originate on the presence of some bacteria, Pseudomonas syringae, that I mentioned the name because they're coming back again. And all the idea at that point was to find out how plants are impacted by pathogens that can attack them by producing freezing under mild cold conditions. So in the 70s and the 80s, biological ice nucleation are the most cited papers of these decades. Then in the 90s, you can see a shift, and this shift is towards cloud and weather models. This is the big race of weather studies, but not as in the 60s and 50s as weather modification, but now the new paradigm is to look at what is in the atmosphere and whether we can model the weather, what it is, and understand what are the substances, natural substances, that are responsible for crystallization of ice and the formation of precipitation. So since then, I would say that precipitation, cloud formation, glaciation, albedo, all of these are the main areas in which ice nucleation has been appearing in scientific publications. But when we look at the biology that appears in the 70s, it's actually unraveling to be extremely complex. We have ice nucleating proteins in many organisms to control the formation of ice with completely different functions. These are just a few of the organisms that produce proteins that actually have evolved to control the formation of ice. We have the case of fish, and maybe you have heard about these polar fish that they live at temperatures that are below the melting temperature, the freezing temperature of water in this fish. And that means that thermodynamically they should be frozen fish, but they're not. They're just moving around, and they have some proteins that prevent the freeze. These are antifreeze proteins. Antifreeze proteins are also very common in insects because all of these organisms that do not control their temperature, they have to really survive big changes in temperature without freezing, because the freezing of the cells typically produces disruptions that lead to death. So beetles and also plants and there are many organisms that produce proteins to stop the growth of ice or to control the crystallization. But there are also a few organisms, including insects, but most commonly I would say are bacteria that have the ability to promote the formation of ice. And these bacteria are actually the plant pathogens that were mentioned before. So these are the bacteria that go into the plants, into the stem of the plants. And when the temperature goes below 2 or 3 degrees below zero, what they do is they produce freezing and they break the cellular structure of the plants and they can feed from them. What was maybe a surprise was that already in the 90s, 80s, but it was finally confirmed, I would say, in the late 80s, and more recently studied with more detail, people found that bacteria that are plant pathogens are actually not only in the ground, but they're also in the clouds, and they're the most efficient ice nucleates in the clouds. So this shows a very interesting and somehow unexpected interaction between the biosphere and atmosphere, in which the bacteria that are in the plants, when there is aerosolization because there is wind, they go up to the clouds, and in the clouds they play a role, not the most important role, in the formation of ice and precipitation. What this graph is showing is the density of particles that can nucleate ice, so how common they are, these particles in the atmosphere, as a function of the temperature at which they are able to form ice. And the particles that are more common, unsurprisingly, are inorganic. So we have a lot of dust, soot, ash. These are they're moving in very large distances. For example, snow formation in California can arise from Saharan dust, and this is quite common. But these particles, despite being in very large amounts, they have crystallization temperatures that are not very high. So that means that you have to have clouds at very high altitudes to really partake on the crystallization by these aerosols. Then there is a whole unexpected, in some sense, type of aerosols that are based fully on biological particles, spores, and these are not particularly good ice nucleants, but they have some abundance in the atmosphere. Pollen, that actually is becoming more and more studied because it can influence significantly ice nucleation, but the most significant because of their concentration and also because of the ability to form ice at the highest temperature, so at the warmest supercooling, at the lowest lying clouds, are bacteria. And these bacteria, for the most part, there are many bacteria, but many of them have the same type of proteins as the one called here Pseudomonas syringae. So this Pseudomonas syringae, what they have is in this elongated body, it has patches of proteins, and these proteins that are attached to the outer wall of the bacterium are able to promote the formation of ice. So what I'm going to tell you today is more about how these bacteria manage to have so exceptional ice nucleation. Now, the uses of these bacteria are multiples, but one of them are for making snow. So if you go to a snow ski resort out of season or a little bit early in the season, what you will find is machines producing snow. And to produce snow, you need three things. You need water, you need air to make them fluffy, and what you need is something that also helps to form ice with very little supercooling. Otherwise, you have to cool down the water to extremely low temperatures. The product that is used commercially is a snow max, and a snow max is nothing else than pellets of dead Pseudomonas syringae bacteria. So they culture these bacteria under conditions of starvation that they produce more of these proteins, and then they kill them, although some people say that they're not all dead. You can apparently recover some alive, and they make these pellets, and this is effectively a fantastic method of making snow. So bacteria are extremely good at ice nucleation, and they have evolved to do so. So one of the questions I want to address today is what makes bacteria so outstanding at ice nucleation? And then what are the lessons we learn on how bacteria make ice, and use that also to search for the most potent ice nucleants that would be substances that can form ice without any supercooling. And that means that as soon as you cross below 0 centigrades, the substance can promote the crystallization of ice. This could be the most potent ice nucleus. In this presentation, I'm going to show only a few of the results that we have been working on this for the last few years, but all the results that we have are based on molecular simulations, and that means that we evolve the system in a computer according to interactions that we learn from experiments and from some theoretical understanding of matter, and then we use nucleation theory, mathematical models, and thermodynamics to provide a unified vision of how bacteria form ice. So when we think about the crystallization of water or any phase transformation, what we're talking about is a first order transition. And by this, I mean that we have a phase, in this case is liquid water, that if we are below the melting point, let's say at -2, -3 centigrades, this phase is less stable than ice, so there is a difference in free energy instability between ice and liquid that favors the ice phase. However, to go from the liquid to ice, what you have is to form a new seed spontaneously of the new phase. And this new seed, although it has more stability because it's ice, it has an interface. And the cost of this interface is what really puts forward between the ice and the liquid a barrier. And this is the barrier that has to be surmounted. And probabilistically, the probability of moving through this depends on how high is the barrier. So when we think about the crystallization of water, the main control of what is the temperature at which ice is going to form, or what is the barrier that we can surmount at a given temperature, is given by the surface tension, the cost of the interface between ice and liquid, and the driving force that is this delta mu in this equation. That informs on how to design a substance or a surface that can promote the crystallization, what would be a catalyzer for the formation of ice. Something that catalyzes the formation of ice is a substance or a surface that decreases the cost of that interface. And one of the ways, and the most common way of decreasing the cost of the interface, is by providing a place where the nucleus can grow and the cost of the nucleus surface interface is relatively small, such that the nucleus can be stabilized and get over the barrier at a temperature that is higher. That means that when we are looking at the heterogeneous nucleation, that is nucleation by anything that is extraneous to water, then the magnitude of this barrier, so how easy it is to form ice, depends on how much the ice nucleus likes the surface. The more the ice nucleus likes the surface, the larger, the more negative the binding free energy of the nucleus to the surface, then the higher is the temperature at which ice can form. And we have used nucleation theory, that is a theory based on thermodynamics of looking at the work it takes for the nucleus to grow large enough that it goes into the new phase, to predict what would be the relationship between the binding free energy, the strength of binding of ice to the surface, and the temperature at which the surface can form ice. And what you can see is that it's a smooth line, and the deeper we go, the more we're approaching the melting temperature. And this poses the question of what is that makes some substances like ice-nucleating bacteria, the proteins of ice-nucleating bacteria, some alcohol monolayers, some organic crystals, so good at ice nucleation. But now what we can do is address the same question in a way that is more concrete, is say, what is that makes these surfaces so good at binding ice? And when we look back at these surfaces of proteins, of alcohols, and even antifreeze proteins that bind very strongly to ice, what we find is that all these surfaces, what they have in common is that they have at least some hydrogen bonding groups, OH groups, that can strongly bind with the OH groups of water. Water is two OH groups, they bind strongly, and the distances between the OH groups in the surface and the distance between the OH groups in ice are sort of close. For example, alcohol monolayers, they have a pattern that looks like a rhombohedral. So it's like a squeezed hexagon. And the distances A and B in this rhombohedral structure are very similar, within a few percent of the distance A and B that you find on the surface of ice. Likewise, the proteins that bind strongly to ice, including the ice-nucleating proteins I'm going to tell you about today, what they have are rows of hydrogen bonding groups, and these hydrogen bonding groups also have distances B and A that are sort of close, within a few percent of the ones in ice. So this is a requisite. It's not sufficient, but it's a requisite of all these particles. If we go now deeper into understanding how do they bind ice at the molecular level, what we find is that these ice-nucleating proteins that are typically very long, and by very long I mean in the molecular scale, they're about 30 nanometer long and about 2 nanometers wide, and then they have groups that anchor them to the membrane. So they're large for proteins, not for humans. And these proteins, when we look at them from the front, what they have are rows of OH groups, here shown in blue, separated by other OH groups by groups that are methyl groups. And if you are into chemistry, you know that while the OH groups are strongly binding to ice, the methyl groups are actually the paradigmatic hydrophobic moiety. That means that these methyl groups try to get away from ice. And the combination of the two, intriguingly, forms a structure that is here shown in orange, in which the water gets around the methyl groups and attached to the OH groups in a way that forms what is called an anchor clathrate. A clathrate is a cage around water, around the hydrophobic molecule, a cage made of water. And this is the kind of structure that these proteins promote at the interface. And it's counterintuitive that you need hydrophobic groups to bind strongly to ice, but this is what the proteins do. So if you expose these proteins to water and you cool enough, the ice forms spontaneously, as shown in this picture that is a snapshot from a molecular simulation. And this ice that grows then propagates and crystallizes the full system. When we started working on this, I was quite intrigued about the role of the hydrophobic groups, because ice is hydrophilic. But what we found by performing mutations of these proteins, by changing the methyl groups and changing the OH groups, is that the strength of the binding of these proteins necessitates actually both the hydrophobic and the hydrophilic groups. And if you start mutating them out, if you remove them, what we see is the strength of binding decreases, it goes towards zero, decreases whether we remove the OH groups or the methyl groups. So we learned that both of these interactions are key to produce the extremely strong ice nucleation ability of these proteins. Other thing that was intriguing on these proteins was that the proteins themselves have a very large, they're very large, and they have a beta helix kind of a structure. And this beta helix, you can see that is sort of rectangle. So they have two flattish sides, and these two flattish sides have a repetition of this motif with an OH group, threonine, something threonine on one side, and the groups on the other side is quite conserved that they repeat also these three groups, and where the blue are OH groups. And it has been proposed before by Peter Davis and co-workers that the two sides may help on promoting ice nucleation, but this was not demonstrated. However, our simulations show for the first time that indeed the side that we considered before and the other side that is less hydrophobic, is more hydrophilic, both of them form ice quite effectively. Indeed, they form ice with the same efficiency. And the efficiency in this case we're measuring as the distance from the homogeneous nucleation, where homogeneous nucleation is where you don't need any help, they can just form ice without the protein. So the side that has hydrophobic groups and the ones that have less hydrophobic groups, they have the same efficiency and is comparable to the experiments. What this indicates is that both types of interactions can produce a very effective ice binding. And we found indeed that there are many ways of having a strong binding of proteins to ice. So there is not a single way. But it also illustrates that in nature you have these two, and this suggests that the two of them are used, because if you have one that is ice nucleation is very effective and the other that is very bad, when you cross the temperature at which the more effective works already is forming ice and the other is never used. So this suggests that the two sites are used together. And it was proposed that these two proteins, you have the two proteins like this, one turns around and now the surface that you see in the proteins is actually a combination of the two sides of the two binding sites. So it was proposed that maybe the proteins in the bacteria may be acting as dimers. Another piece that is known, and this has been known from pioneering work already in the 80s by Stephen Lindo and co-workers, is that these bacteria that they have ice-nucleating proteins, they have a wide range of ice-nucleating temperatures. So you take the bacteria and you start diluting the bacteria to the point that you can reconstruct what is the profile of temperatures at which different bacteria can nucleate ice. And what it became evident at that point is that there is a class that is called class A that nucleates at very high temperatures, -2, -5 centigrades. And then there is a class that is called C, because there is no class B in between, that nucleates at -8, -10 centigrades. Now, these two classes were not assigned, and I'm going to show you soon how do we assign what they are, but it was known already that you have bacteria with proteins and that the larger the arrangements of the proteins on the surface of the bacteria are probably responsible for the highest ice nucleation temperature, and if the aggregates of proteins on the surface of the bacteria are smaller, the ice nucleation temperature was going to be smaller. And this was done in an experiment in which they used radiation with gamma radiation to start destroying the proteins and measure how effectively they could nucleate ice. However, although there was a lot of interest in this area, there were two questions that remained that were not answered, and I'm going to answer today. The first one is quantitatively, what is the relationship between the size of the aggregates and the length of the proteins and the temperature at which they can form ice? So how large they have to be to nucleate ice at a given temperature. And the second is, what are the actual sizes of the aggregates that are included in the bacteria? So what are native sizes? Because you can imagine any, but we're going to see that only a few sizes of aggregates can really exist. When we think about the nucleation of ice, what we have to take into account is that ice nucleation requires the stabilization of ice. And the embryo has to be large enough to be sitting to the larger the temperature at which you want to form ice, the larger this embryo at the top of the barrier has to be. But to have a very large embryo that is stabilized, what you need is a lot of real estate. So small proteins can only form small embryos, and they can nucleate ice with very little efficiency. The larger proteins can do a little bit better. But what we found is that if you just take a protein and you make it longer and longer, you can improve the ice nucleation efficiency by making the nucleus larger, but at one point it saturates. And this tells us that the width of the protein limits the efficiency, and that's why in bacteria, the proteins have to aggregate to produce a much larger and effective ice nucleant. Now, not all the ways in which the proteins come together are equally effective. You have sort of an interference pattern because not all the positions between the proteins can allow you to have a perfect matching of ice between the two. That's why there are very few distances between the proteins that can produce extremely good ice nucleation. And from this, what we learn is that the bacteria must have an exquisite control of this distance in the cell membrane. What we don't know is exactly how are they controlling this. And I'm afraid I cannot tell you today because we still don't know, but we're getting there. Now, using this perfect distance and using nucleation theory fed with the results that we get from experiments, what we can do now is to predict where is the ice nucleation temperature of a monomer. This is in kelvins. A dimer, a trimer, so we can now predict exactly at which temperature they're going to nucleate depending on how many of these you have in the aggregate. And now this poses the question of, first of all, how good are these proteins at nucleating ice? And our analysis indicates that actually these bacteria ice nucleating proteins have evolved to be as good as ice itself. So if you have a piece of ice of the size of the bacterium is equally efficient, so it's truly outstanding. But then this poses the question of what are the sizes that are actually in the bacteria. And to make a long story short, because we are also still investigating this, what we can do is take the experimental data on the cumulative number of bacteria, these ice nuclei are the bacteria, that form ice at a given temperature. This comes from many experiments doing nucleation on many samples at multiple 10 different dilutions. And then we do a mathematical modeling to obtain what is the underlying distribution of the temperatures at which bacteria can nucleate ice that reproduces back the same experimental result. And now that we are confident that we have what is called the underlying distribution of temperatures at which the bacteria can form ice, we can go back to the theory to make the assignment. And our assignment, here shown in blue, is that the main peak, what is called class C for this bacteria, nucleates ice at minus about 8, is a tetramer. So there are four proteins coming together. While the class A, that is rarer, is only 1% of the total bacteria, has 8 mers, so these tetramers coming together, and maybe also 12 and 16. So now we have an idea of how these proteins organize, and we have an idea of the size, and we also can predict the sizes based on the experiments. With that, what we know is that we understand that the bacteria are achieving this exceptional ice nucleation by having small units, relatively small units, that they organize to make larger proteins or larger protein aggregates. And these larger protein aggregates are what really confers the exceptional ice nucleation ability. But it has also been found that in these studies in the 60s that then were sort of discontinued, it was found that many organic crystals that have very simple structures also nucleate ice. And we studied a couple of them, the two on the top. And to make a story very short, what we found is that these organic crystals that also have hydrophobic and hydrophilic groups, the same as the bacteria on their surface, induce a similar ordering of water. And that's where, from that is that they derive the exceptional ice nucleation ability that is observed in experiments. So now, very briefly, because I know I have not much more time, I want to show you that we can actually do better than bacteria, and we can have ice nucleation at higher temperatures. And the concept behind this is to show that if the ice nucleation temperature, so how little supercooling we need, is related to the strength of this binding of the ice to the surface, there is a point at which we can strengthen more than ice interaction with itself and open a completely new regime. And this new regime that we predict using theory says that if we have a surface that is increasingly better at forming ice, it will form ice at higher and higher temperatures like bacteria do. However, if we go to an increase in strength that is even stronger, the binding of the ice to the surface than the binding of ice with itself, what we open is a new regime that is called pre-freezing. And that means that you could form ice even above the melting point. This is the prediction from the theory. So I'm going to show you briefly that we achieve this in simulations. And to show you how to illustrate this in simulations, what we do is we look for something that is an exceptional ice nucleant, and then we make it better. And the exceptional ice nucleant can be the bacteria, but I don't know how to make them better, or something simpler, these alcohol monolayers that were already nucleating ice at -1 centigrade. And what we find is that if we compress slightly the alcohol monolayer, what we're finding is that the alcohol monolayer becomes a better nucleant. We have shown this before. So this is what we do in our experiments in the computer. We take the alcohol monolayer that nucleates ice at -1. This is amazingly good. And then we squeeze it a little bit just by 0.3%. And before you see already at 0.3%, you can form ice above the melting point. So the stable phase is liquid water, but already the surface of these alcohol monolayers have a layer of ice. And you see this in this side view or front view in which ice, shown with different colors according to the layers, is forming on the surface. So we have already ice, and that means that when you are cooling down, because there is already ice on the surface, what happens is that the ice already grows, and this is ice nucleation without supercooling. So these are the most potent ice nucleants. And we studied how do they grow the layers, and you may wonder, is this the end of the end of Earth? We're going to have like ice-9, a substance that crystallizes all the water in the planet. But you should not worry about that, because it can only grow a few layers above the surface, but cannot produce crystallization except below the melting point. So there is no planet catastrophe because of this. So with that, I want to get to my conclusions. That is that potent ice-nucleants bind strongly to ice, and ice-nucleating bacteria have evolved proteins that are extremely powerful at binding ice, and they do this by using hydrophobic and hydrophilic groups in a concerted way that order water. And although counterintuitive from the structure, these are extremely potent ice binders and ice nucleants. Then, although each bacterium is very strong at binding ice, is very strong, and the biomimetic or the biological strategy to produce exceptionally high ice nucleation temperatures is to bring many proteins together and putting them together in a way that we don't understand yet how they're bound together, and by doing that, achieves the full ice nucleation potency. And then I show you that we can do better than nature and nucleate ice without any supercooling, and this opens the possibility, because we understand the physics, to design new molecules that can do that. And there are many synthetic analogs that can do this. So this comes back to the question of cloud seeding. Can we design molecules that are exceptional for cloud seeding? Whether this is a good idea or not, I will leave it open. And although research in this area has been discontinued in the last 40 years or so, still programs in cloud seeding continue. And actually, Utah, that is the second dry state in the nation, has about 8% of the water in precipitation coming from cloud seeding. And there are techniques that have not been yet optimized for cloud seeding, but there have been already studies like this one I'm pointing here, also from Utah State University, showing how a new predicted weather, that because we have climate change and the climate is changing, how this change in climate is going to impact the ability of water, of water capture through cloud seeding in the area of Utah, in orographic cloud seeding. And with that, I leave you with a question, because I'm not endorsing cloud seeding. It's a case of environmental engineering that I think that has to be considered very seriously, but also it puts forward the question of how cloud seeding will impact the water rights in a warming atmosphere. And with that, I want to thank my co-workers, Eugene Chow, Atanu Metia, Arpog Dait, Rajat Kumar, Ingrid de Almeida Ribeiro, and Conrad Meister and his team, the University of Alaska, and the funding, and all of you for your time and questions.
Valeria, thanks so much. That was a fascinating presentation. We have a longer queue of questions that I think we'll probably get to today, but I know that everyone would appreciate if you're willing to drop contact information or other ways of staying in touch in the chat, whatever you'd like to do. There's been some requests to stay in touch, and of course we will, again, this presentation will be has been recorded and will be available on YouTube for anyone who'd like to see it again. So just a few questions. I want to start with one that's about the other applications of this technique. So could this ice crystallization technique also be used for dewatering sludge or finding ways to separate water from other biological wastewater solids?
I have not seen that it's used for that. No, I think that it would be too expensive. What the technique is used for is for separation of biological molecules using ice binding. So you can have molecules that bind to ice and selectively separate these molecules through this method. So otherwise it would be quite expensive.
Just too expensive for that application.
In one side it's expensive, on the other one it's very common. These bacteria are very common, and you can buy this 50-pound Snowmax for not much cost. But I have not seen this used for the application you mentioned. Maybe someone has the idea and they can use it. Yes.
Yeah, great. Well, completely other side of the spectrum, going all down into the details about the bacteria. How is it that the dead bacteria maintain protein structures that are so similar to living organisms so that they actually have the same functionality? How is that possible?
How do they keep it when they're dead, or how do they achieve
this?
Yes. What we conclude is that these proteins, there is no active process involved in keeping the proteins together. We don't know yet what is that brings them together, because our study indicates that the fluctuations between the proteins has to be less than 1 Armstrong. That means that if it's not a covalent bond, we suspect that it's electrostatics. And these proteins have one side that is very hydrophobic, that they can make a hydrophobic bond, and the other sides that are charged with positive and negative charges, strategically put such that they can make these tetramers. But that's why they don't need any ATP or active process to remain in that case. On the other hand, if you take the proteins, dead or alive, and you hit them, you impact the structure of the membrane and the fluidity of the membrane, and they don't come back the same. And this is something that has been known and we have been studying more. So there are irreversible changes you can do alive or dead by heating, not by killing the bacteria, but heating, because it changes the membrane. So the properties of these aggregates are intimately related to the properties and the fluidity of the outer membrane of the bacteria.
Yep.
Yeah.
Is there any evidence for some synergistic interactions between the ice-nucleating proteins and other solutes in the water in this nucleation process?
Actually, we are finding that they are. We still don't know exactly what it is, but there are studies already showing that if you change the pH, it changes significantly, and that's not surprising because biological molecules are quite sensitive to pH, and I think that they probably have evolved to have maximum activity at the pH that is inside the stoma of these plants. That is where they act, where they go and feed. But also that they have sensitivity to different type of cations. And one of the possibilities is that some of the cations and anions are really involved on producing the aggregates. We don't have yet definitive answers on that, but we know that you can change the populations from the tetramers to the larger ones by changing the composition of the solution in a way that we're still trying to understand what it means. But you can move between the two populations, but we don't yet know the mechanism.
Is it possible that these proteins could at some point be catalytic for freezing desalination? Is that?
Yes, they could be used, and actually they can be used for other applications, and people have been using them for a few. I have a survey of whether my talk was engaging. I hope.
Oh, yeah.
They could be used for other applications. And for example, one of the possible uses of the proteins, not the bacteria, because using the whole bacteria may have some issues. The question is, how do you use the proteins and extract the aggregates without breaking them apart? And when you break the membrane of these bacteria, if you don't want to have the bacteria because they're large and also they're pathogens, so these are pseudomonas. If you break the bacterial cell, you only get the tetramers, you only get what is called class C. But these could be used, for example, for cryopreservation, where you want to also not only prevent the formation of ice, but control at which temperature the ice is going to start forming. So they have many possible applications, but for that you have to remove the pathogenicity of the pseudomona itself.
Got it.
Yes.
I think the last question, what's the average crystallate size in ice formed from bacterial nucleation? And what does that tell us about the density of proteins that are delivering functionality?
No, when they start forming, typically, I don't know inside plants what are the larger sizes, what are the sizes. But for example, the typical experiments to construct the curves that I was showing before, I have to stop sharing, yes. But I show some curves of distribution. These curves are obtained by freezing thousands of droplets. Each of them is a microliter, and a single bacterium can completely crystallize the full droplet. So there are two processes. One is to form ice on the bacterium, and then when you have ice on the bacterium, it has to grow outside. And at that point, because the system is already super cool, in principle, a single bacterium can crystallize a large size. So you don't need to have many. You can have extremely dilute. Just that when you dilute a lot, you only capture the ones that are less potent, that form ice at -8 instead of -2.
Got it.
Yes.
This has been such a fascinating talk. Thanks so much. And thanks for your willingness to engage with others that were following along. I do hope, selfishly, being here in Chicago, that this is the last time we'll be talking about snow for a little while, since we're finally in spring, but nonetheless, super engaging talk. I want to let everyone know about our next Third Coast Water Seminar coming up at the end of May 25th at 3 o'clock Central Time. We'll have Dr. Rachel Paretsky from the University of Illinois at Chicago, and we'll be talking about Chicago and Illinois' wastewater surveillance effort and the sewage surveillance work that has been tracking early warnings for COVID-19 in the region. So don't miss that. And as always, please stay in touch with Current. There's lots of ways to stay abreast of new events that we have coming up or programming opportunities. Increasingly, we are shifting to include a mix of virtual and in-person over the year to come, which is something that's been long overdue. We're very excited about that, but also want to highlight a couple of new resources, since we know there's educators and those interested in education on these events. Our Blue EDU resources are live if you're thinking about summer learning opportunities for students at currentwater.org Blue EDU. And then, of course, stay tuned for the launch announcement for Chicago Week 2022, which will take place in early October. So we'll be announcing that shortly and look forward to having many of you attend in the lead up and at Chicago Water Week itself. So thanks again very much for joining us today and always, and please join me in thanking Dr. Molinero. And
Thank
we'll
you.
see you all again on the next Third Coast Water Seminar. Thanks, everyone.
Bye-bye.